we have seen a little gold rush of vaccines. For there are hundreds of vaccines under development, but when among all these things, we should only be looking at vaccines which are close to completion, because at the end of the day, we need vaccine in time. So any vaccine which is theoretically ideal but will take many years to develop actually aren't useful actually, because we won't get it which when we need it. So essentially, typically a vaccine development used to take minimum of 3 to 4 years, but up to decades. And except in lucky situation where you can just kill and give the microbe, for example, toxoid, etc., it may be possible to develop something like that, but testing still takes time. So this is the general. So most of the traditional vaccines used to take a long time. This has changed with this pandemic, because something which was theoretically and in labs being studied, that is the nucleic acid based vaccines, actually have shortened the time to the development of effective vaccine. So let us look at the types of vaccines which are front runners for the for actual this COVID. So the traditional front runner, that is viral vector based vaccines. There are primarily two of them and undergoing phase 3 trials. That is, everyone knows about the Oxford Chadoxon vaccine, and a similar vaccine developed by China, the CAN vaccine, and the Russian Sputnik vaccine is also of similar type. One thing is that why it has come early is that they have basically cut short the trials. After phase 1, they have approved for early use. So it is not actually in advanced stages, they are just taken a shortcut. But in principle, they are similar. What happens in this vaccine is that they have taken a virus which causes common cold-like illness, but it's primarily a common cold-like illness that happens in chimpanzees. It's called adenovirus 5, and this virus can also infect humans, and it is possible that some populations may have this prior immune response to this, which may have consequences. So essentially, they have taken adenovirus 5 and put the spike protein of COVID-19 into it. And the reason this vaccine actually, typically such vaccines take years, but there has been an international collaboration led by the Oxford Vaccine Center, Jenner Institute, where they have been developing a pandemic vaccine for any upcoming coronavirus for many years. So they had tested these vaccines using spike proteins from MERS, Middle Eastern Respiratory Syndrome, and the original SARS, Severe Acute Respiratory Syndrome. And the problem was that by the time they developed these vaccine, all these epidemics had died, and there was no way to test the efficacy of these vaccines. So they already had phase 1 safety data on these vaccines, and the Oxford vaccine in previous studies has shown that 80% of the people develop some antibody response. So basically, we have a moderate effective vaccine which had passed preliminary safety in humans in the case of the adenoviral based viral vector vaccines. There are other attempts at development, but they are all early stage. They were not being developed in advance, and so none of them actually have reached this stage. Then the question comes, everybody is talking about the inactivated vaccines because of the Covaxin in India. There are only two countries which are working on inactivated virus vaccines, China and India. And we are not hearing too much about the inactivated China vaccine, except they are given to some soldiers in China. So basically, what you do with an inactivated viral vaccine is take culture the virus, filter the virus, just inactivate the virus, and give the mixture of proteins to individuals. But the problem, there is a technical problem here. When such vaccines were previously developed against SARS in animals, there were some signs of allergic reactions to it. And in previous studies, when spike proteins were used to induce immunity, they also encountered instances of antibody-dependent enhancement. Antibody-dependent enhancement is a situation where prior existence of antibodies lead to more severe disease. Is anyone here aware of a well-known example of antibody-dependent enhancements, which is clinically relevant and we encounter? Can somebody answer that? I think you should. Anybody knows about well-known antibody-dependent enhancement which we see in India? Dengue hemorrhagic fever. So if you get dengue infections the first time, you just have a viral fever, which may not look like dengue, is just a viral fever. You may have myalgia, fever, everything, and you recover. And since there are 4 types of dengue viruses, if you get dengue infection, you are protected from that particular strain of virus. But if you get infected by a new strain, these antibodies are not able to completely neutralize the virus, but in fact enhance the infection by allowing infection of cells, that is mainly immune cells, which normally do not get infected. So these immune cells actually spread the virus much wider and cause much more severe pathology. What we see, that is the dengue hemorrhagic fever and the severe dengue we see, is actually a result of antibody dependent enhancement of a second infection, where people with prior immunity can have a more severe disease. So the challenge of dengue virus vaccine development was that if you develop a vaccine, it should give equal immunity all 4 strains. And if you have partial immunity and a new and non-immune strain gets infected, you will get severe disease. So similarly, when you have an antibody, it is important that you kill the virus completely. You don't want partial immunity. So the issue of spike protein, this was a question mark. So that is why these kinds of vaccines are not favored in most of the places, and more sophisticated and difficult to make vaccines are being tried. So as far as COVID-19 is concerned, whether antibody-dependent enhancement occurs, we do not know yet. But there are also additional reasons for going beyond these antibody-based. So if you are giving a protein or a killed virus, you get antibodies. There is no doubt antibodies will be produced. The problem is that this particular virus, rather actually all coronavirus, can evade antibodies to some extent. So if you look at how vaccines protect, that is immune system protects against viruses. If you look at it, there are 2 main mechanisms, the antibodies and the cell-mediated, T-cell-mediated immunity. So what the T cells do is they identify infected cells. What happens is when the virus infects the cell produces the viral protein, and these are broken down, and bits of those proteins are presented by antigen presenting cells on the surface, and the T cells can recognize. So the T cells can only recognize virus infected cells. They cannot recognize intact viruses. So T cell immunity plays a role in clearing virus infected cells from the body. Suppose if a virus infects a body, if you can clear the cells fast enough, the infection will not spread, and you can limit the disease, and you can save from symptomatic disease or control the infection. But T cell cannot block infection. On the other hand, antibodies can block viruses and can create what is called a sterilizing immunity, theoretically. That is, if antibodies can effectively block the infection, it can be a sterilizing. That is, you will not get infected at all. But if the virus can evade these mechanisms of sterilizing immunity, it will not happen. So this coronavirus has ways to evade it. So if you have enough viruses, virus can escape and get infected. So you really need 2-step defense. That is, virus will break through and then you need T cells to clear the infected cells. So, and since these killed vaccines and protein vaccines cannot produce effective T cell response. So that is why the other vaccines are preferred. And regarding, there is another strategy to select parts of the RBD as a vaccine. People have shown that they can be protective, but still they are actually much more difficult to make, since it requires protein engineering and mass and purification systems are much more difficult to manufacture than other type of vaccines. So they are actually behind the schedule. So this leaves us with the viral vector vaccines and the nucleic acid vaccines. What the nucleic acid, have you, see, the thing is, nucleic acid vaccines are not very new. More than, nearly 20 years ago, when I started studying immunology, they were being talked about. There were a lot of research, but none of the nucleic acid based vaccines actually had come to clinical use, mainly because there was no such challenge like this where efforts were required. And there are two types of nucleic acid vaccines. The older one, which has been being tried for nearly 2 decades, is the DNA vaccine. DNA vaccine, is the DNA which encodes the antigen, which has to be, we have to inject that, and then it goes inside the cell and produces the mRNA, and then that forms the protein. So the advantage of a DNA vaccine is that DNA is high. DNA means that is not mammalian, non-mammalian DNA of bacterial type. That type of DNA is highly immunogenic, whereas mammalian DNA is not immunogenic. So if you optimize the DNA to have that, to get the right balance of immunogenicity so that it doesn't stimulate too much, then you don't need additional adjuvants. So how many of you know about adjuvants? Nobody knows adjuvants. Adjuvants are considered the immunologist's dark secret, because we say that we inject something, produce antibodies, but if you are giving a vaccine without adjuvants, you won't get anything. It's there things which enhance the antigen presentation and immunogenicity. So, like for example, the things like alum, most of the alum basically forms aggregates of the antigen and therefore increases immunogenicity. And complete fluorescence adjuvant basically contains killed mycobacteria. Basically, people have found that mycobacteria contains many things, including DNA, which actually stimulate immunity. So this DNA itself is a potent stimulator of immune system, provided it is like a bacterial type of DNA. How? That is because there are receptors in our body which recognize pathogens, like it's called pattern recognition receptors recognize the DNA, RNA of microbes through receptors. These are TLR7, TLR9, etc. Whereas TLR9 recognizes DNA, TLR7 recognizes RNA. And when they recognize that, they trigger immune system. So DNA does not require additional adjuvant. It is highly immunogenic, potent, very good, produces potent T cell and B cell immunity. There is only one problem. DNA doesn't get into the cell very easily. RNA can get in, DNA doesn't get in very easily. So there are technological challenges which have now been overcome. So that's a DNA. And another problem with DNA vaccine is that it can integrate into the genome, and it's like doing some genetic engineering. So that is a potential danger. Whereas RNA vaccines are relatively new. Main problem with RNA vaccine is that getting it into the cell is not very difficult using, they don't have to get into the, so it's basically, you don't have to get into the nucleus or anything. You just put it in liposomes, like with the lipid nanoparticles, and they will get inside. So that entry part is easy with RNA, but the problem with RNA was very light. They easily destroyed. There are so much enzymes everywhere in every body fluids, even your hand, everywhere there are enzymes which destroy RNA. So keeping RNA stable was a challenge. So this has been solved by chemically modifying RNA, not the usual, nucleic acid bases. They are modified bases are used, which are chemically stable mRNA. They will not integrate. That is, again, one step is avoided. Once RNA gets into the cell, it starts producing protein, and protein being produced inside the cell is necessary for a good T cell response. So it produces good T cell response. Good. So this is, and again, to develop a vaccine, basically you just need the sequence. So lots of steps in vaccine development is avoided, which is why RNA vaccines are and DNA vaccines are the only vaccines whose work started after the COVID pandemic who are in reach phase 2 and 3 clinical trials. All the other vaccines, they were under development long before and they just tweaked them. In this category, there are two vaccines which are undergoing phase 3 trial in the US, that is Moderna, which most of you are aware of, and BioNTech and Pfizer. So these are vaccines which are highly, so basically the most likely winner in the, if you ask me which is likely to be the best vaccine, most effective, at this point of time, my money will be on the nucleic acid based vaccines for another reason. If you notice, the Oxford vaccine in phase 1 trial, only 80% produced antibodies. So there is the reason for moderate efficacy is that it is possible that some people may have antibodies to the vaccine vector, which will reduce the ability of the human body to respond to it. Whereas these DNA RNA vaccines will not be overcome by pre-existing immunity, but since they are naturally short-term adverse reactions like fever, etc. In fact, most people who have got these vaccines in phase 1 trials have got such reactions. But in a way, it's good. It's a sign that our immune system is actively responding. So short-term adverse reactions are more likely with RNA DNA vaccines, but they are now and all of these vaccines have shown that in clinical trials they produce good antibodies and good. And previously it was believed that the Oxford vaccine needed only one injection. Now we know that it also requires 2 injections. Actually, all vaccines need 2 injections for good immune response. Now, regarding trials. So basically we talk about 3 phases of trials, and these trials actually have different purposes. And so basically all research, whether it's development of drugs or with vaccines, it starts with studies in vitro cell cultures followed by animals. So when you do a test in animals, you have some idea of something major is going wrong, whether the development of whether it works in animals or whether it has major toxicity, you can study in animals. But everything cannot be, animals can't talk, and humans are different from animals. We are another type of animal. So we need to do human studies. The purpose of a phase 1 study is usually to look at doses required and to know preliminary safety. That is, if something grossly out of place, you want to know in the first. Suppose if everybody gets an adverse event, you will know in the first few injections. So phase 1 studies are done sequentially. You get to 1 person, then to 2 person, then you increase the number, and then you look at, and these phase 1 studies are done in healthy volunteers. And since vaccines anyway are done given to healthy people, so you can actually combine phase 1, and phase 1 can become phase 2. Phase 1 is about safety. Phase 2 is about preliminary efficacy. Preliminary efficacy means, in the case of a vaccine, preliminary efficacy is, does it produce antibodies? Does it produce T cell response? And so basically, in phase 2, it's an exploratory efficacy also. You are exploring how, in the different ways in which this is working. If it was a disease, you are looking at which manifestation of a disease is responding to your drug. That's what phase 2 looks at. So a lot of things are studied. You have extensive data collected, lots of lab parameters are tested in phase 2 studies. So you will have a lot of baseline information. And generally, the endpoints used in phase 2 are surrogate endpoints. That means you're not looking at hard endpoints, you're looking at endpoints which can be measured in the lab, like neutralizing antibody, T cell response, etc. And then obviously the phase 2 numbers are larger, but you have, and there is no, there are a lot, you might see a lot of slides where people give fixed numbers, but there is no such fixed limit on the phase 1, phase 2, phase 3, etc. It depends on endpoint. If you are looking at things which are going to be given in very large number of people like vaccine, you generally have larger number of people in trials so that you can recognize very rare side effects. Suppose you have a side effect which occurs in 1 in 1,000 people, you won't see that side effect until you give several thousand people the drug. So it is the how common a particular manifestation is, how much small effect you want to see, that determines the sample size. There is no fixed sample size. And finally, you need phase 3 studies. Phase 3 studies will tell us. Are you everybody following me? Hello? Need questions? Hello?
अच्छा अभी क्वेश्चन पूछ रहे मैंने.
Okay. In phase 3, you want final proof that it is working. In the case of a vaccine, you expect to show that this vaccine prevents the disease. Showing antibody is not enough. Maybe antibodies may not work. Maybe T cells may not work. Although so final proof is in, so if you are giving a drug, you want actual measurable differences like mortality, measurable things like number of cases reduced. So in a phase 2, you can have, you have some idea of number, that is you, if you want to see, if you want to show that. Hello? Can't hear you.
This is a clinical trial.
Hello? Yeah, I can't hear clearly. Hello? Okay. In a phase 3 trial, basically you have the number of cases. So the size of phase 3 trial depends on the infection rate in the population. That is, if you do it in an area where there are large number of new infections, then you can do a trial with small numbers and you will get results early. So what you do in a phase 2 is you give the vaccine and test the antibody response. Whereas in a phase 3 trial, you give the vaccine and wait for COVID cases to occur. Then you compare the number of people who get COVID in those who got the vaccine versus those who did not get the vaccine or they got a placebo vaccine. And then you, from the number of cases in the placebo vaccine is actually some other vaccine unrelated to COVID has been given. And then you compare, you make an expected number in the vaccinated group and see the actual cases are significantly less than the expected. And you have a prior target how much it should reduce. Ideally, you don't want anyone who got the vaccine to get infected. That means that be 100% protection, but you don't expect. So in this, the FDA, for example, has kept a target that there must be at least 50% effectiveness for vaccine to be approved. So that is where we stand. And the front runners, obviously, at this point are the adenovirus-based vector vaccines and the predominantly RNA-based vaccines from Pfizer and Moderna. And there is something else I need to bring up. So besides antibody-dependent enhancement, there is another potential side effect we would like to see. Suppose if you are giving a vaccine first and somebody gets COVID, you hope to get some protection. What happens if you had COVID and then you take a vaccine? So this is a gray area. One potential thing that can happen is that now we know that after COVID, some people have long COVID. Whether this is a body's reaction to it or virus is persisting in all cases of long COVID is unclear. But we know that at least in some patients, virus can be persistent because there are some case reports where patients who had COVID presented with vasculitic manifestations. And when they biopsied, I found there is persistent virus in the endothelial cells, that's in the blood vessels. So suppose if there are areas of the body where the coronavirus can hide from the immune system and stay, and if you give a vaccine and the immune system gets activated, then they can clear the virus. That's one possibility. And when they clear the virus, it can be asymptomatic or it can be symptomatic. So Something like a immune reactivation syndrome can theoretically occur. There are no such reports, but that is a possible danger we need to be aware of. So this is the reason why phase 3 trials are important. Suppose we indiscriminately start using without knowing safety data, like some people are proposing, we might have a scenario where you may get these adverse events. So finally, when guidelines for use are to be given, you need to know can it be given to everybody, or should it be given to people who did not have it, or should it be given to people who didn't have antibodies. So these are things, unknown factor, we should be careful about before we get too excited about vaccine. But overall, the vaccine should be useful, because vaccine induced immunity can be much stronger than the natural immunity. How? We know that when people get infected, the antibodies to coronavirus last only about few months to maximum 6 months. And some people actually clear the virus without even produce, up to 5 to 10% don't even produce any antibodies. And once, and then people have been reinfected also. Why is this happening? Now we know what is happening, because in the presence of COVID infection, there is a defect in the production of antibodies. There's a defect in the production of good antibodies. What is a good antibodies? Antibodies which are long-lived, antibodies which are high affinity to the target. And such antibodies are produced in a specialized part of the lymph nodes called germinal centers. Germinal centers are like the gymnasiums of lymph nodes, where there's the antibody responding B cells proliferate, and where they compete for binding the antigen, and the ones which cannot compete, they die off, and the winners are selected essentially. So essentially, it's a highly accelerated evolution. It's micro evolution which is happening over days, in which high affinity antibodies are formed, and they lead to the production of plasma cells which produce antibodies for the rest of your life. These germinal centers do not form in COVID. This has been established, and some of the mechanisms are also being worked out. So we know that in a COVID infection, germinal center formation is impaired, and so which is why the antibodies are not that good and they are not that long lasting. So, but if you are giving a vaccine, this limitation is not there. The mechanisms which inhibit the germinal center formation are not there. So the antibody response from vaccines may be better than the natural infection. It can even be longer lived than but we have to prove that it's too early. You're just giving the vaccine, time will tell. So so there is a clear case. There is, even though we have to be cautious, there is a clear benefit from having a vaccine, and a disease which has such extensive economic and demographic implications. We need a vaccine quickly, and let's hope this gets approved as soon as possible. Any questions?